Welcome to the Outline Printing course. In the last lecture, we have discussed how to what is vascularity and what are the anatomy of vascularity, what are the different structures present there, and also the what are the ways the angiogenic remodeling can happen and all these things. So, in this lecture, we'll discuss further about the bioprinting of vascular networks. So, first we'll discuss that how to generate a blood vessels with patent lumens. That means how to generate open unobstructed lumen by bioprinting technique. So there are a few techniques by which people trying to do that, but coaxial bioprinting is a significant step towards that direction. Here, generally coaxial nozzles are used. Coaxial means when there are concentric nozzles, when there is a inner core and then the outer there is outer channel for in the same nozzle where two different materials can be simultaneously pumped or extrude through these two compartments, like the inner compartment and the outer compartment. That basically bioprinting this kind of concentric nozzle has a very special application where two different materials, either the one biomaterial can be or biowing can be extruded through the inner core nozzle, and the cross linker can be extruded through the outer channels. In that case, the cross linking of the biowing will happen as soon as the extrusion happen. But suppose if I extrude the biowing through the outer concentric channels, outer channel, and through the inner core, if I pump or feed the cross-linking solution, then what will happen? Cross-linker, then the cross-linker will cross-link the biowing that is coming out as a tube-like structure, and inner it will be filled with the cross-linker. So we can get a tube-like structure filled with the cross-link inside. So inside the structural patency can be very much intact because of the cross-linker present. The cross-linker will cross-link the channel from the inside out, right? So that's what is actually there are a few groups who are working on the thing and who have actually worked on that are similar that thing where the concentric nozzle has been used for that same purpose, when the feed nozzle was with a biome cross-linker that is distributed through the inner tube, and the outer tube contains the desired matrix biome encapsulated cells, and during printing the outer and inner tube dispense the material simultaneously, so resulting in the outer tube with a matrix biwing and the inner cylinder with the crosslinker. So the crosslink field tube will be developed, and then because the crosslinker is presence of crosslinker in the inner structure, inner inside the tube, so it will cross-link the structure biwing from the center luminous center to the outward. So maintaining the patency during printing. So Gao et al. Actually they have combined this bioprinting of coaxial vascular conduits with to create microchannels for nutrient delivery. What have they have done? They have printed a that they have used a concentric nozzle extrusion where they have used sodium alginate as a biwing and loaded mouse L929 fibrous cell lines, and then they have also filled the inner core of that concentric nozzle with that is connected to the CSL2 solution feeder, and then they have used that the extrusion coaxial bioprinting concept to generate that sodium alginate tube like structure that is filled with the calcium chloride solution. So the calcium chloride solution here is a cross linker for the sodium alginate, and by this way the sodium alginate biowing that will get solidify and will get a lumen, so that open lumen tube like structure, and that is filled with that L929. They have used a little higher sodium alginate concentration to so that the structure structure retention would be better, and the structure is solidified. First thing is the structure is solidified, and also the structure is stability can be maintained, also the mechanical property can be good. So in this work they have shown the possibility of using this kind of a open lumen tube like structure for bioprinting. So this is very significant advancement towards the development of a uh, freestanding vascular blood vessels, big blood vessels. Also, in the subsequent work, they have also encapsulated the endothelial cells, the human progenitor cells within that vascular luminal structure, and then the tissue derived. Also, they have loaded this one another drug that has some effect on the vascular, the angiogenic tissue development. So that with the disulfide structure and ACL alginate composite hydrogen. So they have made a composite of disulfide plus alginate and use the same concept of coaxial bioprinting, by which the tube-like structure has been developed, and that was filled with calcium chloride solution, and then that can be used as a blood vessel for further applications. So they have also done some in vivo studies in that work. Please, please go through this literature. Due to copyright issue, I have not presented the image and other details here. But please go through this typical literature for better understanding of this work. This is a very significant work in that direction. Now we'll discuss some other concept that is called indirect printing with sacrificial material. Till now we have seen how to generate the blood vessels by direct 3D printing of the hydrogel material. Now we'll discuss how sacrificial materials can be used. Sacrificial material can be used to generate these kind of structures by indirect 3D printing. Because in indirect 3D printing, with this concept, what we can do, we can create the channels. We can create that blood vessel like structures in a tissue block or in a cell laden hydrogel, and then that channel can be used as can work like a channels embedding in a tissue bath. Because what happens, the sacrificial materials are here most of the time. These materials are either thermo reversible material, where they can be used at certain temperature, and when we change the temperature, then they will become liquefied, and then that can be taken away, so that they leave the channel, and channel where again the media can be perfused through that channel. So there are different sacrificial materials, various different sacrificial materials are being used. Something like gelatin, pronics, all these things have been used as a sacrificial material thing. This also, this first work, that is Miller et al., they have developed a sacrificial meal based on sugar. Sugar, you know that sugar can be melt, extruded, and then that can be the strands, freestanding strands can be printed. So in that particular work, what they have done, they used sugar as a material, sacrificial material for printing the network-like structures. And then when what happens, these network-like structures, after printing, the cell and hydrogel is poured over there in that sugar network, printed network. And then what happens, then the gel, then the cell and hydrogel was allowed to become gel or solidify. Then the sugar, then when they have again washed that thing with water or media, then the sugar is actually can be dissolved away. So the after the sugar dissolved away, they leave channels within the tissue block that is prepared by the cell and hydrogel. So in this way, then these channels can be again perfused with media, and also they have also these channels can be perfused with endothelial cells. So the endothelial cells can go and bind to the inner lining of that channels, and by this way a tissue with vascularized tissue construct, where the vascular tissue is embedded within the tissue blocks, can be developed. So this work has shown the possibility of using or generating pre-vascularized tissue, where the vascularized tissue bed, where the for vascularity, the channels are created, and the channels are again can be endothelialized with by seeding endothelial within this channel, and then this can work as a vascular tissue construct. So this is a very significant work where they have shown the possibility of developing a vascular tissue construct with sacrificial printing with sacrificial material. But this work is limited in terms of, because when you are using cell-laden hydrogel, the most of the time the cells will be homogeneously dispensed or homogeneously suspended within this hydrogel. So we can get a structure where one or two types of cells can be homogeneously suspended in that hydrogel. So and then we can get the tissue with that. But we cannot control the positioning of this hydrogel in that you, you cannot control the positioning because it's a molding technique. First the sugar template was 3D printed, but then the cell and the hydrogel was poured in that using a mold. So it's a molding technique in that way, in the where the sugar template was used as a cast to print the whole to print that customized tissue construct. But in molding technique where we cannot position the cell and hydrogel based upon our interest or based upon the application, so we cannot control the positioning of the cells. Also the hydrogel cannot be positioned well. So basically we will get a homogeneous tissue construct, heterogeneous tissue construct with different cell density, different cell presence of cells at different locations. That is not possible in this world. So Koleski et al. Has also worked on a similar concept of using sacrificial material as a printing of sacrificial material for creating the channels, and they have used pluronics. Or sorry, there is there's spelling mistakes. It should be pluronics. So they have used a pluronics as a sacrificial material to create the channels with the tissue within the tissue bed. So the pluronics is a material which at over 20 degrees centigrade it's like a gel like material, but below 20 degrees it's like liquid material. So what they have done, they have used pluronics to print the channels that can be sacrificial materials, but simultaneously they have also printed cell-laden hydrogel along with this thing. And as a cell-laden hydrogel they have used gelma as a material, as a binding material. And so through one nozzle cell-laden hydrogel, that is the cell-laden gelma, was printed, and with another nozzle the pluronics was printed as a channel, as per the vascular network design. Then they have used photo cross linking, because gelma is a photo cross link material. Photo cross link for to solidify the structure or to make the structure, so this thing gel. And then they reduce the lower the temperature to less than 20 degrees centigrade, where the pluronics is like a pluronics liquefied, and then the pluronics can be taken out easily from the by suctioning. So this work they have shown that by using pluronics as sacrificial material, a cell-laden hydrogel with embedded channels can be created, and these channels can be further, they have endothelialized with by perfusing endothelial cells in a media through the channel. So the endothelial cells go and bind to the innermost layer of this inner lining of the channel, and then the uh, tissue blocks with embedded microchannels are possible. So this is this is the work. Please go through this literature. First for Miller et al. They have published this work in Nature Materials, and Koleski et al. They have published it in Advanced Materials. They have also worked further on the same thing, where they have printed a 1 centimeter thick vascular osteogenic tissue construct. What they have there, they have used osteogenic progenitor cell like stem cells for creation of the printing of the structure. Stem cells was embedded or encapsulated in a biwink, and similarly the pluronics was used as a for creating the vascular channels. And then they have printed both these things simultaneously to come up to fabricate a 1 centimeter thick vascular tissue when the sacrificial material is removed, and then the channels are embedded, channels are intact. So then they circulated media through these channels, and it was found that then the and when optimum differentiation factors were given through the channels, then the emissions printed within the cell and hydrogel could differentiate into osteogenic lineage like towards osteoblast, and they could develop a osteogenic tissue construct. Also, it was found that the cells present near to the channel survived much better than the cells present far than the channel. Or so, a perfusible thick vascular tissue construct was developed in this work. Please go through the literature, that is patient and PNS. I could not add images from this because of the copyright issue from those papers from the literature. But please go through the literature; you will understand much better about the work they have developed with sacrificial materials. Similarly, branching of vascular network is another important requirement, because in the body, as I discussed in the earlier chapter, the vascular network, they generally present in as a branch. So this branching is very much important for vascular network. And with one such study published in around 2019, Norte et al., they have worked on a similar concept where they have used cell spheroid and also extrusion bioprinting. They have used to generate this bifurcated tissue tubular structures. What they have done, they have printed with high Hubeck HVMSCs, my fibroblast and SMCs were printed in a small tube multilinear tube-like structures, where they have used here agarose strands as a supporting material, where the agarose strands are there, and in between the agarose strands these cell spheroids are printed, so that the cell spheroids cell spheroids are printed. So when I print this agarose cylinder, agarose rods that in a perpendicular in a parallel fashion, and in between that agarose rods, if I put my spheroids, and then again agarose rods are printed, so the spheroids, they will fuse with each others, and then they will be a cylindrical tissue kind of structures can be very well generated. Now if I put another agarose cylinder in between these things, in between the spheroids, I can come up with a tube-like structure. Then the tube-like structure can be developed with this with the agarose cylinders as a supporting material. Now I can create bifurcation there also by using some other agarose cylinders and putting the spheroids in that fashion. So by doing that, the spheroids will fuse to each other, and they will mature, and a vascular network will develop. When the agarose is removed by solubilized, the agarose is removed, that time the tissue will be easily developed. However, in vivo vascular branching is not limited to horizontal. In this case, they have mostly worked on that, and they have shown the promise of creating the vascular channels or creating the branching of tubular structure in the same plane. But in case of in vivo or in native tissue, the branching can also happen in different in the no, it's not limited to the only one horizontal plane. It can be of the different vertical planes, so that kind of thing. So there are more strategies required to generate this kind of structures. And as I earlier mentioned, that the printing of vascular channels within the embedded gene by that the 3D bioprinting on in support bath concept, that can be very well used for to generate that kind of structures. So let us now understand to the what are the things required for what are the requirement for a larger blood vessels. Like the first thing is for larger blood vessels, anastomosis is a fact where the joining of the artificial blood vessels, or in this case the printed blood vessels, synthetic blood vessels, with the existing blood vessels is very much important, where when these two connected seamlessly, they can connect with each other. That is called anastomosis, so it's connecting to the existing one. So that is very much required for synthetic blood vessels, because they need to attach to the existing blood vessels for circulation of blood through that thing. So that is very much important. So anastomosis is the very first requirement for the thing. Then functionality post transplantation, when it is transplanted with that existing blood is connected, that then the synthetic blood vessels should be functional, so that they should carry the blood without thrombus formation, or without leaking of the blood, or without getting blocked. So those things are very important. So post functionality post transplantation also the, because the this thing, the blood synthetic blood vessels has to be, when it during if it has to be connected with the existing one, then suturing is the way to how we can connect these blood vessels with this thing. And if the suturing is successful, then that blood vessels can, but the means that can be functionally active, and it can also uh, the blood can flow with this thing without any problem. So the other requirement is there should not be any thrombus formation at the suture site, or there should not be any blockage at the suture site. That is another important thing. So that suturability of the vasculature in synthetic vasculature with the existing one is very much important. And then other thing is that synthetic vasculature should have enough mechanical property so that it should satisfy the suture retention and burst pressure. Suture retention, when it is connected, sutured with the existing one, there should not be any tearing and wearing of the synthetic blood vessels during this thing. And also when the blood is flowing through this thing, because if it is connected to the artery, then it will go the systolic and diastolic pressure it has to withstand, and that time it should not be able, it should not just burst away. So it should be have a typical strength so that it can withstand that pressure. Sufficient intactness of the endothelium to prevent thrombosis. Then other than this thing, there should be endothelium grow over the synthetic one during in vivo. So that the endothelium is what happens when you connect the synthetic blood vessels with the existing one. That thing, the tissue is generally the endothelial cells from the existing one, actually they migrate and they populate the synthetic one. So that synthetic one should support that endothelium cell migration and creation of a endothelial lining within this thing, so that if the endothelial lining is fully created, then it will prevent thrombosis, and there will be no blood clot within the blood, within that synthetic blood thing. So that this is that endothelium, a intactness of the endothelium within the synthetic blood vessels is important. It should support that. And also there should be high patency rate to support occlusion free circulation, because suppose if the when the this thing when the synthetic blood vessel is implanted in use or in some patients, then this should not cause any kind of blockage or leaking or this thing. So that means they should be able to do this thing very nicely. Thank you very much for joining this lecture. In the next lecture again, we'll meet in the next lecture.